Hey guys, it's Miss Vigo, and today we are going to learn about another process of cell division called meiosis, and we will see how it differs from mitosis. We're going to learn how gamete cells are made and discuss what gamete cells actually are. Um, So remember from chapter 1 when we discussed the characteristics of living things, and we said that one of the characteristics is that all cells reproduce. We briefly mentioned the two types of reproduction, asexual and sexual reproduction, where asexual reproduction uses the DNA from only one parent to make an offspring. This flatworm that you can see over here on the screen uses asexual reproduction, where it creates an exact copy of itself because there is only one parent and one set of DNA. Therefore, it is producing a clone. In sexual reproduction, both parents provide the genetic code or DNA to create the offspring. Therefore, the offspring is genetically different than the two parents. Okay, so let's take a closer look at those two kinds of reproduction. So when you think of asexual reproduction, you should think of that DNA is only coming from one parent. And remember, we talked about this last time during division of bacteria. Remember, the division of bacteria is what we call binary fission. Now, since these are unicellular organisms, this would be considered asexual reproduction because it is making a new organism and it is an exact copy of itself. Other processes that are considered asexual is budding from a plant, where you can take off part of a plant, such as a branch from a plumeria plant, and plant it somewhere else, and it will continue to grow into a new organism with the same exact DNA as the parent plant. Also, regeneration is considered to be a type of asexual reproduction. For example, this flatworm that we just talked about, you can actually cut this flatworm, or this particular species of flatworm, in half, and it would regenerate in both parts and make two new flatworms, both with the same exact DNA. Okay, now, if you think about mitosis is kind of like sexual reproduction because it makes daughter cells that are identical to the parent cell. So we start off with one body cell, so it goes through mitosis and we create two exact copies of that cell. So that's kind of like asexual reproduction, except it's just making body cells. So there are some advantages of asexual reproduction. First, you can make lots of offspring really fast. You don't need a partner compared to how long it takes to make a human baby. Compared to how long it takes to make a human baby or an elephant baby, which is almost 2 years. So the advantages would be fast reproduction and not needing a partner. Now, there are also some disadvantages of asexual reproduction. Since the genetic code is coming from one parent, all offspring will be the exact same. This makes it so that the species will not be able to change or adapt to the environment, which is always changing, right? So we know that the environment is always changing, so it doesn't allow for them to change or adapt. So if a disease starts to spread, all the organisms are the same, therefore no one is immune and they all will die out. So it can wipe out one whole population. Now, in sexual reproduction, when the DNA comes from both parents, it makes it so that the offspring are different from each other and genetically different from the parents. The advantages of doing sexual reproduction is that it allows for variation within a population because everyone will end up being different. So, if you think about everyone on the planet, think how they are all different. This provides a foundation for evolution. Or organisms as a group to change over time. This allows for more adaptations to occur in our always changing environment. So having a variety in the group allows for species to change over time. So if a disease starts to spread, not everyone is susceptible, and this is why variety and diversity is actually a very good thing. Okay, so let's talk more about sexual reproduction using a sperm and egg cell to make a baby. So you can see we have an egg cell and a sperm. When they combine, they make a baby. Now, we've talked about mitosis and how mitosis creates exact copies. So if two sperm and egg have the same number of chromosomes, think in humans we have 46, and mix the 46 chromosomes in an egg with the 46 chromosomes in a sperm cell, we would get a baby with 92 chromosomes. This is not a human baby. Extra chromosomes in humans can actually cause very serious birth defects. So, if the baby is going to have 46 chromosomes, we need to figure out a way for cells, for egg cells and sperm cells to have half the number of chromosomes, or 23 chromosomes. And this would allow for a 23 chromosome egg to mix with a 23 chromosome sperm cell to make a 46 chromosome baby with the correct number of chromosomes. Okay, so meiosis. So how do we do this? How do we actually make cells that have half the number of chromosomes? We can't do mitosis because we always end up with the same number of chromosomes. So what we need to do is take a cell and divide it twice into 4 cells to get half the number of chromosomes. And meiosis is this process. Meiosis is the process that is used to get half the number of chromosomes. So you can see here, we start off with one cell, divide once, divide twice, and then we end up with half the number of chromosomes. Okay, let's just skip over this. Okay, so before we talk about meiosis, we first need to talk about some vocabulary. So think about your cells and how you have 2 copies of each chromosome, 1 copy from your mom and 1 copy from your dad. And we call these homologous chromosomes, right? And because they have 2 copies of each type of chromosome, we call that diploid. Remember, we have 23 chromosomes. We have 23 types of chromosomes, so 23 pairs, or 23 types, and 2 chromosomes of each type. So we have 2 number 1s, we have 2 number 2s, 2 number 3s, and so on. All body cells are diploid cells, and often we write it as 2N. So we have diploid, or we call it 2N. Because there is 2 chromosomes for each type of chromosome, therefore it is called 2N. So all body cells are diploid, and the word for body cells is somatic. So all body cells are somatic cells, soma meaning body. So when we refer to cells as somatic cells, that means they are body cells which are all diploid, or 2N. So muscle cells, skin cells, liver cells, heart cells, they're all somatic cells. And remember, our homologous chromosomes are the chromosomes that are pairs, right? They're not exact copies, but they do contain the same type of genes, and they're the same size, same shape, and we get one from my mom and one from my dad. Okay, now the other type of cells are called haploid cells, and haploid, think about the word haploid. It kind of sounds like half. So haploid has half the number of chromosomes, or 1N. Remember, if you think of diploid, di meaning 2, so 2N. So half of 2, 1. Okay, therefore haploid cells are 1N because they only have 1 copy of each chromosome, and all sperm and all egg cells are haploid cells. And we call these sperm and egg cells gamete cells. Remember, we want them to have half the number of chromosomes than body cells, so that when we put them together to make a baby, the baby will then come out with the correct number of chromosomes, which is 46. Okay, so let's compare a little bit in general mitosis and meiosis. So remember, in mitosis, when we have a new cell, we make two genetically identical cells to the parent and also to each other. And we are making what we call 2N cells or diploid cells. This is the way we make more body cells. So we are making somatic cells or body cells. And this is used by organisms to grow bigger, from one cell to trillions of cells. We use it to repair injuries, skin cells, make more skin cells. We use it to replace worn out cells. And so when we're doing mitosis, we are doing it to make cells that are the exact same. Now, when we do meiosis, we are going to end up with 4 cells that are genetically different from each other and also from the parent or starting cell. Okay, and remember now, we want to have half the number of chromosomes because we are making sperm and egg cells. So meiosis will make 4 cells, or 4 haploid cells. And remember, these haploid cells have half the number, so they are 1N. And these make the gametes, the sperm and eggs. And this is used in sexual reproduction. And so you can see here, we start off with the 2N, we start off with the diploid cell, and then we'll end up with 4 haploid cells, all 1N, having half the number of chromosomes. So what kind of body cells are going to be doing meiosis? Because if you see here, we start with a haploid cell, which is a somatic cell, the 2N, as we just said. And this will go through meiosis. And that makes 4 haploid cells, or gametes, right? So these would be our gametes, or haploid cells. Well, if you think about the body, think about what system contains the sperm cells or egg cells, and that would be the reproductive system. So it will be cells in the testes or ovaries that will actually be going through meiosis, not necessarily a lung cell or a skin cell. But also the ovaries and the testes, they can still go through mitosis, but they are also the ones that go through meiosis in order to make the sperm cells and the egg cells. Okay. So what's going to make meiosis different? So basically there are 3 things, or 3 different phases that are actually different in meiosis than mitosis. Number 1 happens in what we call prophase 1. And prophase 1, we say 1, remember, because in meiosis they are going to be dividing twice, so they go through the cycle 2 times. And the first time through, we always put a Roman numeral 1 next to each phase. And the second time through, we always put a Roman numeral 2 next to each phase. And that way we can tell which division we are talking about. So it's not necessarily confusing when you're talking about the different phases and which division, the first division or the second division. So in prophase 1, something that happens is what we call synapsis and crossing over. This does not happen in mitosis. And the second thing is called segregation and independent assortment. And this happens in anaphase 1 of meiosis. And then the third thing that is different is that we are going to be skipping the second interphase. So it does not go through S phase again, and we do not copy that DNA again. So the cell will only divide 2 times, but only copy the DNA 1 time. So let's look a little closer at these 3 things. Okay, so remember we talked about homologous chromosomes, where we have 2 chromosomes that carry the same traits. One you get from your mom and one you get from your dad. these are found in your body cells. So in meiosis, the parent cell is a somatic cell, or a diploid cell. And in prophase 1 of meiosis, these homologous chromosomes will find each other and form a little group and stick together. And that's called synapsis. That's called synapsis. So now we have 4 chromatids. If you can look at this picture, so we have a homologous chromosomes, right? So here's one chromosome, here's another chromosome. And for each chromosome, we have our sister chromatids, right? And these sister chromatids are exactly the same in one chromosome. And in this chromosome, these sister chromatids are exactly the same. So we have 1 chromatid, 2 chromatids, 3 chromatids, 4 chromatids. And this group of 4 chromatids is what we call a tetrad. Tetra means 4. So during prophase 1, synapsis occurs, and the homologous chromosomes will actually find each other and actually intertwine with each other and form a tetrad. And this is different and does not occur in meiosis. Okay, so now that the chromatids have been intertwined with each other, they can actually switch their DNA. And this is what we call crossing over. Because these little piece of genetic material from the maternal or the mother chromosome will actually switch the genetic material from the paternal chromosome, paternal meaning dad. So you can actually notice the color change over here. You can see that some of the orange chromosomes switched over to the blue chromosome, and the blue chromosome switched over to the orange chromosome. And this is what we call shuffling the DNA, which allows for genetic variety and for the sperm or egg cells that are made to be genetically different from each other. Okay, so let's go ahead and watch a video of crossing over.
Crossing over occurs during prophase 1 of meiosis, when homologous chromosomes align together. Once the chromosomes have paired together, breaks will occur in non-sister chromatids at identical sites. The non-sister chromatids exchange broken segments, which are then sealed together by specific enzymes. Crossing over breaks up old combinations of alleles and puts new ones together in pairs of homologous chromosomes. This process increases genetic variability.
Okay, so let's go back and talk about what's going on with our chromosomes. So remember, homologous chromosomes are the same shape, same size, and carry the same exact type of gene, but they are genetically different from each other because, remember, one comes from your mom and one comes from your dad. So you can have the green-eyed gene from your dad and the brown-eyed gene from your mom. So it's important to know that they are not exactly identical, but they do contain the same type of information. Okay, so let's look what's going to happen to the DNA after crossing over. So if you look at, we can have the mother chromosome here and the father chromosome here, or the one from the mom, one from the dad. And before crossing over, you can see that the sister chromatids are the exact same. Remember, they are the exact copies because they're in S phase. That is when the chromosome is being copied. So you can see that they are exact copies. Obviously, they are different because this is the mother and this one is the father, but these sister chromatids are going to be the exact same. And you can see these little colored bands here. These colored bands are what represent the different genes, so the different hair color, eye color, height, and so on. And let's just say, these ones up here represent eye color, so we have green eyes from mom or blue eyes from dad, and down here represents, the hair color. And so down here will represent red hair coming from your mom, and down here would represent brown hair coming from your dad. And so after crossing over occurs, you can see all over here, the sister chromatids are now actually going to be different from each other because they're actually going to be splitting or switching at different sites, or at the same site. And they are going to be now actually different from each other. So this allows for rearranging of the DNA. And now the sister chromatids are not going to be identical to each other anymore. Okay, and so this is what happens in prophase 1 after synapsis occurs, and then they trade their DNA, actually mixing up that DNA and putting different combinations of DNA together. So this, okay, so they will be different. Okay, so and now instead the mom could be passing on brown hair that was traded from the dad, or red hair that was traded from the mom. Okay, and so the number 2 thing that we said, that another way meiosis is different from mitosis, is the separation of chromosomes during anaphase 1. And this is called segregation and independent assortment, which is another way that the genetic material is shuffled, which then again allows for genetic variation. So let's go ahead and watch a movie on segregation.
The chromosomes of all organisms contain regions called genes. Each gene determines a particular trait of the organism. For example, one gene in pea plants determines seed color. Another gene determines seed shape. A diploid cell has 2 matching sets of chromosomes. Matching pairs of chromosomes are called homologous chromosomes. Homologous chromosomes carry the same genes in the same locations. Since all diploid cells have homologous chromosomes, each cell carries 2 copies of each gene. However, the 2 copies are not always identical. The 2 copies of the gene may specify 2 different versions of the same trait. Seed color, for example. One version of the gene on one chromosome may specify green seeds, while the version on the other chromosome may specify yellow seeds. The different versions of a gene are called alleles. The cell also carries 2 different alleles that determine seed shape. One allele for wrinkled seeds and the other allele for smooth seeds. Now consider what happens in meiosis. The chromosomes replicate. In metaphase 1 of meiosis, homologous pairs of chromosomes move to the middle of the cell. In anaphase 1, the homologous chromosomes are separated and pulled to opposite ends of the cell. 2 haploid daughter cells are formed. When the homologous chromosomes were separated, the alleles on those chromosomes were also separated. The allele for yellow seeds is now in the right daughter cell, while the allele for green seeds is in the left daughter cell. This process of allele separation is called segregation. The alleles that determine seed shape have also segregated. The allele for wrinkled seeds is in the left daughter cell, while the allele for smooth seeds is in the right daughter cell.
Okay, so segregation occurs in anaphase 1, and remember, A for ana, A for apart. So we're going to separate or pull apart our homologous chromosomes, and one partner will go to one daughter cell, and the other partner will go to the other cell, and that is what we call segregation, almost kind of sounding like separation, segregation. Okay, so you see the homologous chromosomes are going to be the ones that line up together, and during anaphase they get pulled apart, so one chromosome will go one way and the other chromosome will go the other way. So we have one from the dad going one way and one from the mom going the other way. And so you can see here in segregation and crossing over at the very beginning during prophase, we have that crossing over of the chromosomes. And then what happens is after that they get separated from each other. Okay, so then segregation occurs, and you can see the chromosome with half of the father's chromosome with half the mother's chromosome, and the mother's chromosome with half the father's chromosome. Then eventually gets split again, because remember we're making cells with half the number of chromosomes, and you can see that they are all genetically different from each other. Okay, and the other thing that mixes up the chromosomes is independent assortment. And remember, during metaphase 1, the homologous chromosomes line up in the middle, Remember, M from meta, M from middle, and then they get separated in phase 1. Well, this is important in the way that they line up because the mom's chromosome goes to one side, and then the dad's chromosome will go to the other side. However, not all the mom's chromosomes are going to line up on one specific side. They're going to line up differently and randomly, so that there's many different combinations of how the homologous chromosomes will line up. Think about how many chromosome pairs we have. There's 23. So those 23 pairs can all line up differently. So for example, we can have 15 chromosomes from the mom on the right side, and 8 chromosomes from the mom on the left side, and the exact opposite for the dad's chromosomes. So all these different combinations also will allow for genetic variation, and this is what we call independent assortment. So you can see in this picture, there's different possibilities. Okay, let's just say there's 2 different types of chromosomes, right? Remember, the red we will get from mom, blue from the dad, and so what happens is they can all actually line up on one side and get split, or they can line up on different sides and get split that way. So then this cell would end up with half of the dad's chromosomes and half of the mom's chromosomes, and the same with the other cell, but just different chromosomes. Right? So you can see, even just with 2, there's a bunch of different combinations, and think about how many we have. Right? We have 46 chromosomes, so we can actually end up with a lot of different combinations. And this is what allows for, genetic variation. This is why you look genetically different from your brothers and sisters, even though you guys actually have the same parents, because all the different combinations of the sperm and egg cells. Okay, so, here's just another part of the independent assortment. Remember, back here is showing you 2, here is showing you 3 different ones. And like I said, there's many, many different combinations that can occur, and this is why you actually look different from your siblings and the parents. Obviously, you guys are going to look very similar, but this is why you guys don't look the exact same. Okay, so here you can see this animation. This animation is basically showing you what's going on in meiosis. Remember, we end up with those 4 cells starting off with a diploid cell, copying that DNA. Crossing over is going to occur, independent assortment, and then you end up with 4 cells that are all different from each other and all containing half the amount of DNA that we started with in the parent cell. And you can see all of them are very different from each other. Okay, so let's recap the differences in meiosis. We have crossing over, segregation, and independent assortment. Right? So crossing over, segregation, independent assortment, and these are all ways that meiosis actually results in genetic recombination, or what we call genetic recombination, where all daughter cells are actually genetically different from each other and from the parent cell. Okay. So let's actually go through these phases, starting with mitosis. Remember, in mitosis, we start with the D1 phase, which is the beginning of interphase. So we have D1, S phase, where the DNA is being copied, then D2 phase, making those cell parts, getting ready for optimal division. Then we go through prophase, metaphase, running up in the middle, anaphase, getting pulled apart, telophase, getting that nucleus back, and then cytokinesis, which is actually the splitting of the cytoplasm. So here you would start with a somatic cell and actually end with a somatic cell, or start with a diploid cell, end with a diploid cell. Start with the cell and end with the same exact cell, right? So you're ending with the same exact DNA, same exact copy. Okay, so now meiosis actually starts the same with D1 and with a diploid somatic cell, and then it goes through similar phases, right? S phase, where the DNA is being copied, D2, making those cell parts, then going to prophase, metaphase, anaphase, telophase, and cytokinesis. But But now that the cell is going through a second split, instead of going back to interphase, we're going to go back to prophase. And so remember there's two divisions, and so remember we use that Roman numeral. So for the first part, we are going to call that prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1. And then for the second division, it's going to be called prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2. Okay, and that allows us to tell the difference between which ones we are going to be talking about and during which actual split. Okay, so another thing is that you can see that after cytokinesis, it doesn't actually go back to G1 S phase or G2. We're actually going to be skipping that interphase and going to prophase 2. So after cytokinesis 1, we go straight to prophase 2, then phase 2, and phase 2, phase 2, cytokinesis 2. Okay, so let's go ahead and watch a video of what's going on in meiosis.
In prophase 1, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses, and they exchange segments by crossing over. By metaphase 1, the nuclear membrane has disappeared, and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole. In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the two sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes.
Okay, so let's walk through the phases of meiosis real quick. So remember, interphase 1, we have that G1, S, and G2 where the DNA is being replicated. The cell's actually doing its job. And then during G2, the new cell parts are going to be made. So after interphase 1, we're going to start the division process, going to meiosis 1 or prophase 1 of meiosis, where the chromosome pairs are going to be, or the homologous pairs are going to be pairing up and forming that tetrad. The spindle fibers are going to form, the centrioles will form, and then the spindle fibers will attach at the centromere of the chromosome. So metaphase, M for meta, and for middle. The homologous chromosomes are going to pair up and line up in the middle. And then in anaphase, A for ana, A for apart, homologous chromosomes are going to be splitting apart from each other. And then telophase 1 and cytokinesis, where the nucleus then forms and the cells separate. Okay, so then we're going to go through a second division. And so you can see here with our first division, they still have the same number of chromosomes as the parent cell. Okay, and so then our second split, we start with prophase 2. Remember, we're skipping interphase 2. We only go through interphase 1. So we're going to start with prophase 2, where the nucleus is going to disappear. And the spindle fibers form again, the centrioles contract. And then we're going to go through metaphase 2, where the chromosomes actually line up in the middle. and anaphase, where the chromosomes then get pulled apart, and then telophase and cytokinesis 2, where the 4 daughter cells will split apart, and they will all be genetically different from each other and contain half the number of chromosomes. So we have our 4 haploid cells, and remember, they will all be genetically different from each other. Okay, so let's compare and contrast the different phases in mitosis versus meiosis. You can see here on the left side we have mitosis, and on the right side we have meiosis. So let's compare what's going on in the different phases of the different processes. So we can see in interphase, remember interphase, the cell is doing its job, the DNA is spread out as chromatin, the nucleus is going to be visible, and then during S phase, the DNA gets copied, and after that DNA gets copied, the cell's going to go into G2 phase, where we're making new stuff for the cell and growing and getting ready for cell division. So that's interphase of mitosis, and guess what? There is no difference in interphase 1 of meiosis. It's the same exact thing that's going on. Okay, so then in prophase of mitosis, the DNA is going to scrunch into chromosomes, the nucleus will disappear, the centrioles of spindle fibers will appear, and the same thing is happening in prophase 1 of meiosis. The only things that are different is that the homologous chromosomes are going to pair up, and then synaptic and crossing over will occur in prophase 1. Remember, that was one of the differences between mitosis and meiosis, so not too much that you need to remember. And then in metaphase, remember, and for meta and for middle, for mitosis, the chromosomes will line up in the middle. And then for metaphase, it's actually different because the homologous chromosomes are actually going to be lining up in the middle. So you can see this is just a straight line of chromosomes, but here we have our homologous pairs that are actually lining up together. And then with anaphase, A for ana, A for apart, and mitosis, the chromatids are going to split, but in anaphase 1 of meiosis, chromatids are going to stay together, but the homologous chromosomes are going to split apart. So you can see that the homologous chromosomes are splitting apart, still containing their chromatids. Okay, and then telophase in mitosis, T for kilo, T for 2, you can now see the 2 nucleus forming and 2 nuclei forming. The DNA is going to be spread out then again as chromatin, and the centrioles, spindle fibers, will disappear. And this is the exact same thing in meiosis. So you don't have to really need to remember too much in that one. Okay, cytokinesis, the cytoplasm is going to split into 2 cells, and this is going to be the same thing in meiosis for cytokinesis 1. And then interphase. So what's going to happen in mitosis after the cells go through that split? Remember, it's going to go back into interphase, right? So we just call that interphase. So it's not going through another division, it's just going through that cell cycle. So in interphase, remember, the DNA is spread out as chromatin, the nucleus is going to be visible. If we look at it under a microscope, the DNA gets copied in S phase, and then in G2 phase, it's going to be making new cell parts. So the only difference is in meiosis, we're going to actually skip into phase 2 so that the DNA does not get copied. Because remember, we want to end up with half the amount of chromosomes in our 4 daughter cells. Okay, so this is actually the easiest part of meiosis. Basically, what I'm just going to show you is that all the second phases of meiosis are going to be the exact same phases as mitosis. So in prophase 1, the DNA is going to scrunch to the chromosomes, the nucleus will disappear, centrioles, spindle fibers will appear, and like I said, same in mitosis. Metaphase, the chromosomes are going to line up in the middle, same as mitosis. Okay, in meiosis, metaphase 2, chromosomes line up in the middle. In anaphase, the chromatids will split and move apart, and the same thing's going to happen in anaphase 2. And then telophase, the nucleus, 2 nuclei will appear, the spindle fibers will go away, the centrioles will go away, DNA then spread out as chromatin, same thing in telophase 2. Okay, Same thing in cytokinesis. Cytoplasm will split. Cytokinesis 2, the cytoplasm will also split. Okay, so here's just an overview of how meiosis is different from mitosis. And again, we have those homologous pairs that actually match up. Synapses and crossing over will occur until phase 1. And then segregation and independent assortment happens in anaphase 1, creating genetic recombination. And then we skip into phase 2. Don't copy the DNA twice so that we can produce haploid cells. Okay, so these are the 3 main things that meiosis is very different from mitosis. Okay, so now we need to talk about how making sperm and egg cells are different. So making a mature sperm is what we call spermatogenesis, and they mature by making their long tail that we call the flagella. Remember, we learned about that before. They have to be able to swim to the egg. So if you look down here, you can see that we start with a diploid cell, and it goes through 2 divisions to make 4 functional daughter cells, all haploid. So every time the testis cells goes through meiosis, 4 sperm cells that are capable of working will then be made every single time that those testis cells go through meiosis. Okay, so if you compare a sperm cell to the size of an egg, a sperm cell is a lot smaller. So if you look at the head, you can actually see that there isn't a lot of room. And this is because sperm cells only provide the DNA. This is because we want the sperm cell to be very small and lightweight so that it can swim really fast and go really far in order to reach the egg. So basically what happens is the dad only provides chromosomes, and most of the cell parts or the starting material, the nutrients, the organelles, and all the molecules that provide the starch for the baby is actually going to be coming from the egg cell or the mom. Therefore, we need the egg cell to be big in order to fit all of those needed resources for the growth. This is why meiosis is going to be a little different from making egg cells and making sperm cells. You can see that our sperm cells is a lot smaller than our egg cells. So our egg cells are going to contain all of those necessary materials that we need for growth. Okay, so when a mature egg is made, this is what we call oogenesis or oogenesis, because an oocyte is the egg cell. Okay, that's what we call the egg cell, an oocyte. And you can remember that from the cell trials case study that we did. So during meiosis for an egg cell or oogenesis, what happens is the cytoplasm will actually be splitting unevenly. All the cytoplasm will be given to one cell during both processes of cytokinesis. So you can see in the first split, right here, in the first split, all the cytoplasm is going to go to this cell, and no cytoplasm, or very, very, very little cytoplasm will actually go to this smaller cell. So basically, all the cytoplasm, all the nutrients, everything, all the cell parts are going to go to this one side, and only the DNA that was copied, remember, because we split the DNA, is actually going to be going to this one little cell. And then the same thing will happen again in meiosis 2 or in the second split. The little cell will divide again and basically only split the DNA. So you can see here, the little cell will split again, only splitting that DNA, whereas the bigger cell will divide and the cytoplasm will go to one side, go to one cell and create only one mature egg cell, and then end up with one smaller cell that only has that DNA. So you can see we end with only one mature egg, and then there's 3 smaller cells that only contain DNA, and we call these smaller cells polar bodies. So during oogenesis or meiosis for an egg cell, only one mature egg is being made, whereas in spermatogenesis, there are 4 mature sperm cells being made every single time going through meiosis. Okay, so why only make one egg? Well, that one egg actually has all the nutrients and necessary resources to be healthy and grow properly, because the egg cell is what is actually going to be providing those necessary resources, not the sperm cell. The sperm cell is only going to be giving half of the DNA, and basically the mother or the egg cell is going to be providing all those nutrients and all those cell parts. Remember, the mitochondria you only get from your mom, you don't get from your dad. So all of that is going to be coming from the mother, and only half the number of chromosomes are going to be coming from the father. Okay, so let's talk about these polar bodies. So what do the polar bodies actually do? Well, what happens is that they are actually quite useless. So what they will be doing instead is they will actually get digested. And think about which organelle plays a really important role in digesting unwanted materials. Lysosomes. Remember, lysosomes, they contain those enzymes that will digest anything that we don't need in the cell. So these small polar bodies will actually go through programmed cell death, or what we call apoptosis. And this is actually for the good of the organism. They are giving all the good cytoplasm to one egg for the good of the baby. Okay, so this is actually a good thing, right? Since all the small polar bodies are basically sacrificing themselves to give one egg actually all of the resources and materials needed, so that we end up with only one mature egg every single time a ovary cell, or that somatic cell, will go through meiosis. We're only going to end up with one mature egg rather than 4 eggs like the spermatogenesis, where spermatogenesis ends up with 4 sperms. Okay, so that is the end, and hopefully it wasn't too long, and I will see you guys soon. Have a great week. Hopefully you enjoy your time off. See you soon. Bye.